Great, thank you. So today I'm going to be talking to you guys about heterogeneous mixtures of dictionary functions for finite approximate Koopman closure. This is a story that Igor's heard many times, but by popular request, I've had multiple people say, please explain this to me. I want to understand how this works. And I'm going to throw in lots of killer applications. Unfortunately, I'm going to let Joel down dramatically because I don't think I can top the control algorithms that he was able to present yesterday and the videos that he presented. But I do have some cool movies for you. So I'll wake you guys up when you arrive there. So first and foremost, I'm going to introduce an algorithm that was published, oh my goodness, in 2016. We submitted it to ICML. It was summarily rejected. The reviewer said that LSTM and RNNs and everything else could beat anything that could come up with Koopman operators. And so I went ahead and benchmarked faithfully an IPy notebook and showed how Koopman operators actually did a great job forecasting on all of the models that we built for this paper. The long story short is that we published it in a controls venue because at that time, controls didn't know better to reject Koopman operator work. And then it was funny because by the time we published it, it was already cited like 70 times as just an archive paper. So it was this interesting finding. What we found in that paper was that if you take a perceptron, or a multi-layer perceptron, I should say, a deep feed-forward network, the simplest network you can think of, with just standard activation functions, exponential linear units or rectified linear units, and then you use those, the outputs of those neural net as dictionary functions, to essentially do EDMD, but you try to learn the Koopman operator and simultaneously learn the parameters of the neural net as you're training to solve an interesting regression problem that this type of algorithm does extremely well. So as an example, a system that we introduced to the community as a benchmark at the time was not a very popular system, but we popularized it, I think, is the glycolysis network. And the glycolysis network consists of 7 different states. This is an abstraction of a complex biological pathway. But basically, you can think of it as a standard 7th order nonlinear differential equation with fractional nonlinearities here, here, and there. Lots of bilinear terms as well. So it's a standard system. And the question that you would often ask is, given an initial condition, could your algorithm predict 300 steps into the future without diverging from the correct response? All right. This is a necessary condition, not necessarily sufficient for Koopman function space invariance. So that is if you had some observable and you map a Koopman operator on it, right, it should ideally stay in that function space. It shouldn't map somewhere else. If you're using a bunch of dictionaries, it should be the case that if your Koopman operator acts on those dictionaries, that you're still in that same space, right? If you're not, then a necessary condition would be that your prediction is going to be terribly off, all right? And so here's the comparison from that paper where we looked at how EDMD did, and we didn't just do one EDMD algorithm. We tried all kinds of different dictionaries because we thought maybe we're just messing up. So at the time we looked up Nathan Cutts and Steve Runton's papers and tried to try every single collection of dictionary functions under the sun. And what we found historically was just that all of the predictions were good for the first 50 to 100 time points. And then as you went further out, because we were doing n-step forecasting, so call back to Will's talk, right? But n-step forecasting is not just about one-step minimization error, right? It's about doing n steps forward in time. So you really have to have learned the phase space. You have to have learned the function space properly. And what we found was there was a huge phase shift and also instabilities in the simulation. Now, when we did it with deep DMD, what we found is we actually were able to learn the dynamics of the system. So that is, given a new initial condition that had never seen before during training, it was able to predict exactly how the system would respond. So this was surprising to us. It was interesting. We also were a little bit aghast that eDMD didn't work so well. At the time, analytical DMD had not been invented, but nonetheless, this was the finding. And so this raised the question. So I was telling Harry, it took about 6 hours of coding in TensorFlow, because that's what we did it in back then, and about 30 minutes of actual execution on your Python notebook to do all those experiments. But then it took us about 2 to 3 years to understand how it was so successful. So how does deep DMD learn dictionary functions that satisfy or approximately satisfy the Koopman equation? And so all of that was published this year, finally, in May of 2025. And it's in a paper that is similar to the title of this talk, but in particular, it says, why do deep Koopman operators work? Why do deep Koopman operators work? So If you like this material from this talk and you want to read more about it, there's a very long paper that has this, and you can just search this on Google. Why do deep Koopman operators work? And it'll come up. And the basic starting point of this is that we need to satisfy the Koopman equation, and we want to do it in an approximate way because deep DMD is a numerical method. And so we say that given a dictionary function that is a vector of components, so to be precise here, psi1 is one dictionary function, psi2 is another dictionary function, and psi-n is another dictionary function. You stack them all together to form a vector, right? So we say that this achieves finite epsilon closure or finite closure with order epsilon error when there exists a projection of the Koopman operator, k in Rn by n, and an epsilon greater than 0 such that d psi dt is equal to k psi plus epsilon of x. And we say that psi achieves uniform finite approximate closure for some set R when it achieves finite closure with a bound on that error, this approximation error, that is true for all x in that R. All right. And that's the ideal. That's what we want, is to be able to have a collection of dictionary functions and that there is a bound on that error. And so a little bit more than about this. So analysis of closure by bounding error, or by trying to figure out how big this error is going to be, is a really important way to approach this problem. What is closed with respect to what? So first and foremost, we want the span of the dictionary functions to be closed with respect to the action of the approximate Koopman generator. So if we have a function v that is in the span of psi, then we want it that when k acts on v afterwards, it's still on the span of psi. And then approximate closure with uniform boundaries, again, is to say that it's true across all x in some set. And the question, of course, is given a dictionary that potentially can be tuned, how low can we get b to be? And this is generally challenging because there's very few restrictions on f of x. And so really you want to be able to start with a set of dictionary functions that are universally approximating functions, but you also want them to be very effective at universal function approximation. So this is one clue as to why neural nets are very good at doing what they do, because they are actually really effective at searching across function space. I'll show you videos with that in a second. So an example then of a dictionary function that's commonly used but that doesn't satisfy this property is monomial dictionary functions. Monomial dictionary functions like x squared, x cubed, x to the fourth, etc. Have suffered this problem that if you write down x dot equals x squared, and typically you're writing down some dictionary, some global dictionary of the form x squared, x to the n, then what happens is that when you look at the derivative of x to the n as required for looking at d psi dt, right, and the nth component, you're going to get dx to the n dx dt. That's going to give you this emergence of this new term, x to the n plus 1. So this then means that when you're looking at this expression, now you have to deal with an error term, and that error term is going to be, this property is not going to be satisfied. The error term is going to be large. All right, and so this is generally one of the reasons why we think that eDMD, just vanilla eDMD, does not work. It doesn't work because you're not taking into account the fact that you want to keep this property true. So while we get the regression part of our model to be very good, to regress the vector field, we're not taking care of the subspace dynamics, okay? And by subspace here, I mean the space of functions spanned by the dictionary, all right? We want to be staying in that space. We don't want it to move out of that space. Okay, so now here's a picture of what happens, coming back to our example, of what neural nets learn with deep DMD. And you can see here plotted on this essentially 20 different functions overlaid, all right? And each one is corresponding to a different final node in the output layer of the neural net. Okay, these videos are running forward and backwards, so you can see what happens towards the end of the simulation, and especially towards the beginning of the simulation, where most of the action happens twice. But basically, at the very beginning of the simulation, you can see there's a very rapid, sudden shift as the neural net is quickly searching from what our initial guesses of what the function should be, and as it gets the error, it's basically adjusting and correcting towards functions that it likes. And you can see the functions it likes are functions that look like logistic functions or sigmoidal functions, and then a couple that look like distorted or asymmetric radial basis functions. Okay? So the takeaway is that deep neural nets learn mixtures of dictionary functions. All right? Not just one class. And we, as human beings, typically write one class of dictionary functions down. We're like, I'm going to use the Chebyshev polynomials today, right? But they like to learn mixtures. So how do you combine different dictionary functions in a way that's congruent, that's compatible? That's tricky, all right? So I'm just going to show you a couple more examples, and some of these visualizations might be a little bit more close to home. Oops, I didn't mean to do that. So first and foremost, for the toggle switch, you can see the dictionary functions here plotted that are learned by deep neural nets. And secondly, for the Van der Pol oscillator, here are some of the functions that are learned. These ones, this one actually looks like a combination of radial basis functions, so we haven't studied that kind of function yet. But both of these here all have elements of the RBF. And we can formalize the nonlinearities that this DPMD is learning by writing it down as a logistic curve. And logistic functions have a mathematical form that will then allow us to hypothesize if we can create mixtures of dictionary functions and then effectively run an EDMD algorithm that utilizes the same combinations of mixtures as a neural net, then the ansatz, the educated guess here, is that that combination of dictionary functions is going to perform as well as deep DMD. And if so, in so doing, then we've sort of cracked this puzzle of what's behind the black box, okay? All right, so of course we have to do this in multiple dimensions. So instead of just writing it as a scalar, we're going to write it as a multivariate function. It's going to take multiple logistic functions and then basically acts like an AND gate. So it takes multiple inputs and then it produces an output signal that is on at certain regions. Okay, but I think of these like tabletop functions or hill functions, however you want to call them. We call them conjunctive logistic functions because they are combining multiple inputs at the same time. Same is true for your radial basis function. You have your standard expression here for a radial basis function. And we're going to be considering here now products of radial basis functions that are scalar, so that way you get something that looks like a multivariate Gaussian, okay? Okay, so those are our two dictionary functions that we're going to be analyzing. And the question is, how do they play together well? How do we satisfy the subspace invariance property approximately, okay? So, the closure proof is long and arduous. I'm going to show you one half of it because otherwise I won't have time to show you the killer apps. And I'm in the biology session, so I feel obligated to show some biology examples. But basically, we would like to have some starting point, and a reasonable starting point is that we chose enough dictionary functions so that we can approximate the vector field of the original system, x dot equals f of x, very well. All right, because if we don't even do that, then it's like, what's the point of studying subspace invariance? So we're going to assume that x dot equals f of x can be approximated very well with our collection of dictionary functions. And this is not a hard thing to do. It's basically you populate enough RBFs, populate enough of the logistic functions in your dictionary that eventually you can capture all the different granularity of the function, right? And so that we can say then we'll write as a safe assumption, we'll write the f of x is equal to the summation of a bunch of conjunctive logistic functions and then some multivariate radial basis functions, okay? And I realized I forgot to start my timer. So I just want to see how I'm doing at time. Okay, good. All right, so the question, of course, is how low can the bound go? And so the step that's tricky now is to look at the Lie derivative. We'll look at what's happened as we compute the derivative d psi dx, and we multiply by dx dt. We're getting new nonlinearities now. In the case of monomials, that introduced a new term that wasn't part of our dictionary. That was the problem, right? So what will happen with these functions, right? And so that's our strategy, is to look at this particular step. Let's go ahead and zoom in on that step, all right? So one nice thing that one of the findings from this study is that bilinear functionals approximated by this product process of d psi dx multiplied by dx dt, those terms go to 0, or they are represented by linear expansions of the original basis. Why is that true? I can show it to you graphically. I think graphically is honestly the quickest way for you guys to understand this. So if you imagine having two conjunctive logistic functions from your original set and you multiply them together, what happens to this guy right here, right? Everywhere where this term hits a point where this has 0 support, or support with a 0 value, that just goes to 0, right? So everywhere where both terms are exactly on, all right, then basically these terms are reflected and repeated And there might be some scaling because this may not be perfectly at 1, but nonetheless, this is modulo a scaling constant, the same functional form. So when you multiply two logistic functions, two of these dictionary functions that are learned by neural nets, right, together, what happens is that you get back the same function. And it's the function that happened to have a corner or a threshold that was higher in the space of this set of dictionary functions. And you can actually prove and show that if you have a collection of logistic functions, you can define a partial order. And then from that, you can then actually figure out if you multiply, say, two functions together, the one with the higher order is going to be the one that is the approximate term. Okay. Now, what happens for RBFs? RBFs are simpler to think about, especially when the peaks are not aligned. If the peaks are not aligned, then what happens is that your terms basically cross annihilate because the support or the peak for one spot, right, and the support and peak for another spot basically don't coincide. And so everywhere where it's 0 in one place hits another, and so that then results something like this. Okay, but I've addressed the nicest scenarios. What about the boundary cases? The boundary cases are where you see the error. So you can imagine that if you have gradually rising logistic functions, functions that are not particularly steep, not approximating piecewise continuous functions, all right, what happens is that when you get this guy multiplying with its gradual rise, another function that has gradual rise as well, all right, you can actually get these error terms associated with new nonlinearities propagated. So the best way to implement this is with sharp RBFs, steep RBFs, and steep logistic functions, because then your cutoffs are very, very precise, and also the region of the phase space where you see this sort of error pick up or build up is very small. Okay, so that's the first allusion to what we were talking about earlier this morning about piecewise continuous functions. That seems to be the case with neural nets. They seem to learn sharp, nearly piecewise continuous functions, all right? And it seems to work out in the sense that it then reduces the error in the subspace invariance, okay? Okay, now, once you have this insight, okay, now you understand that you can then take a EDMD approach, all right? Almost EDMD. You still have to figure out where the centers of all the RBFs are going to be, the steepness parameters. So for that, we use stochastic gradient descent, just a standard optimization algorithm. And then you can compare what happens if you run deep DMD versus SGD, stochastic gradient descent. And you can see the rate of convergence is almost comparable. All right, the rate of convergence is almost comparable between these two. All right, so here is SGD plotted in red, and blue is deep DMD. All right, and you can see deep DMD has a head start. But in the long run, over 2,000 epochs, they are comparable. They reach comparable levels of performance, okay? So this is evidence to us that we have understood what deep DMD is doing. Have we understood it perfectly? No, there's still a lot of open questions. I'll come back to that in a bit. But certainly this is a way to think about this. So one of the things that we're thinking about going forward is all right, so we have parameter comparisons here. So deep DMD uses 3,900 parameters. The mixed dictionary uses 995. So you already have a way to get all the benefits of deep DMD without having to, implement a neural net. Not everything has to be on a neural net, right? So, and of course, all the details are present in this paper. What we're thinking about going forward is now that we know how to analyze subspace error approximately, how we analyze model approximation error. Now, how about when we think about control inputs and the notion of control subspace error? So as we're trying to represent the control subspace with dictionaries of nonlinearities, like how do all of those terms come into play as we think about trying to do data-driven control? And so that's a question that I will address at another time at the Koopman operator workshop number 3. We're in the biology session, so I want to talk about a killer app number 3, which is biological applications of Koopman operator methods. And I hope to convince you today that we should do more Koopman operator theory in biology. I think we can really change the fate of living cells. Okay, here we go. So, can we train a living cell to respond to a new chemical? This is a hard question. This is a very hard question in bioengineering. And in Koopman operator theory, with Koopman operator methods, you can do it in 3 to 6 months, all right? That's the takeaway, all right? So, so here's a picture of cells that have been labeled with fluorescent proteins. You guys are all familiar with that. And it's a bacterial cluster that has biofilm genes that detect when they're starting to get dense and crowded, that turn on. And there are 2 different genes that are regulating this. One is red and one is green. The specific functions of these genes are actually not very well known, but we do know that they're correlated. Their activity is correlated with biofilm regulation. And you can see here that cells naturally are able to detect when something is happening in the environment. When they're crowded with their buddies, they know to turn on certain signal, right? So bacteria naturally detect environmental cues such as crowding or the scarcity of nutrients. Can we harness their genetics to detect new molecules? I have some new chemical I found at Home Depot, or I found at some shelf, or Sigma Aldrich, and I want to be able to detect that compound. Can I use biology to do that, right? This is a hard problem. See, before Koopman, the way to do this was with a PhD student, all right? I hope you guys don't mind. It's the last session. I can use a little bit of humor to keep you all awake, okay? So this is, the sad part is if I was presenting to a biology audience, nobody really laughed. They'd all just nod their head approvingly. So, okay, but chemical of interest is here. All right, this is an organophosphate compound. It's a Nazi compound. It's a pesticide associated with maintenance of cabbage plants. And typically what you do if you were to try to solve this problem, you say, I want to detect this new chemical, is you go read papers. Why? Because you wouldn't know what proteins or what adapter molecules could respond to that chemical. And it was hard to dream up some protein structure. To this date, it's still pretty hard, right? Even with AI methods. So what would happen is then they would go to the literature and hope to find a paper in which someone had tangentially touched on the subject and maybe found some genetic markers. All right. Then after that, they would clone and clone and clone, test, and then usually fail. And then they would go back and read more papers. They'd test another hypothesis and test another hypothesis. That was the design, build, test cycle that biologists would do to try and get a new detector or a new sensor for a chemical of interest. And this usually takes 5 years, hence 1 PhD, all right? So they finish the job, they have a new sensor, they publish their dissertation, and then the rest of the field reaps the rewards, all right? And basically you can see papers like this. I've given an example from 2013 and an example from 2021, I want to say, 2022. And the first example, basically they were using mining, using bioinformatics algorithms. They were searching in the literature for proteins that could detect specific compounds that were orthologs of a specific sequence that we already knew worked. So they're kind of relying on initial hypothesis and saying, what other proteins are like this? Oh, they detect other compounds, but they have similar function, right? The second one is this paper right here, which was just published recently, is new, engineering an oleic acid-induced system for a Halomonas and E. coli and Pseudomonas. So they actually managed to get it to work. They extracted it from some exotic organism, and then they tried to domesticate that process. So it's a long, arduous process. You have to find the relevant proteins, isolate the protein, test each individual for ligand protein specificity, identify the promoter that transduces that protein ligand binding into an actionable signal, and then finally test the new biosensor. So you can imagine why this takes 5 years to do. It could be depressing. Now enter the world of Koopman. What do? So in our approach, we said, let's take this problem, which we know has been visited many times by synthetic biologists and people have struggled. Let's see if we can use data-driven methods to actually identify biomarkers in the genome that will respond. Well, how do we do that? We need to apply a persistently exciting signal. So we hit our cells with this new compound, our little pesticide, and we just watch them respond. And we took time series measurements. We conducted an experiment that most biologists would say is foolish, frankly, right? But at the time, DARPA was willing to fund it, so we said, let's do it. So what we did was we sampled the cells every 15 minutes, and just watched how all of the mRNA dynamics evolved as a function of time. Now, I was telling my friend in the front here that when we did this, we saw a lot of unnecessary data. There was a lot of modes that were not relevant. So we projected down to what you're seeing here is about 600 genes. But there are 4,000 genes, but most of them aren't doing anything. Those are already filtered out. In this representation, what you see here is the fold change response, red indicating a high level response, blue indicating a low level response of each of the genes as you move across time in response to the new chemical. So what do you do with that information? Well, of course, you try to extract the modes, you try to plot the eigenvalues, right? And so that's what we did. And then as soon as we did that, we had this question of like, what do we do with these modes? And at the time, I didn't know enough Koopman operator theory to understand what I could do with the modes. But nonetheless, we needed a way to be able to extract the modes the genes that actually matter the most. Now, when we looked at our responses, we had a lot of modes that frankly were just like, it looked like they were just consequence of machine learning. They didn't really tell us anything. But there were 3 modes in particular that were interesting. The stable response modes, well, this was a flat line, the mode itself. But the genes that comprised that dynamic were genes that basically averaged out. So it was like an averaging function here. Then there was an unstable response mode, which was basically all the genes that were running off in fold change and never seeming to come back down. And then there were genes that were plotted here that didn't seem to have a response at all. They were flat lines, okay? They made it through the filtering process, but they're still relatively unresponsive, okay? So now the question was, how do we identify the genes, though, that matter for biosensing? And for that, we actually turned to observability maximization, because we realized if we take everything from this mode, those genes might be correlated. They may all be on the same pathway. We don't want that. We want 15 to 40 different genetic markers that are all doing different things that give us a very diverse, rich set of signals that we can use. So we said, why don't we try to find a way to extract the states of the cell that are like orthogonal in some sense, that they act in different directions and respond in different directions as a function of time. And so for that, we use the observability gradient, because that allows you to partition your response subspace into the different orthogonal modes that coincide with each individual gene. We then plotted the participation factors for each of the genes, and what we got was an exponential distribution. That is to say, a small group of the genes actually did most of the work in responding to this new chemical. We then took those genes, and just to give you a sense of this needle in the haystack problem, here is a genome depicted. It's fully annotated, but the annotations are so thick and so dense. There are so many genes on this genome. It's 4 million base pairs long, all right, that when you zoom in, this is what it looks like, okay? Each of those little blue arrows is a gene, okay? DMD plus observability maximization gave us these targets, all the red lines that you see on this genome. It said these are the ones to look at, all right? Now, if you just ask me as a biologist to find those, that's why it takes 5 years and a PhD student, and so many don't go on to become professors. But basically, we did it this way, and then we said, why don't we go ahead and build it? It's been about a month of simulation analysis. Now it's time to build it. So it took about 2 months to build. So by month 3, we're at the stage where we have our genetic markers, and we plotted the response, the baseline response here. You can see each of these are responding. And now what we do is we take the promoters from the genome, follow this blue arrow, drop it in front of a fluorescent protein, okay? And then after we do that, we then put that on a plasmid. We take the plasmids and transform them into bacteria. Yes, we did do these experiments. Yes, I'm a biologist as well. And then we have each individual strain. So we have like one marker that we identified goes into one well. Then another well, and a totally separate well and compartmentalized, is a different strain that has those different plasmids. And then we see how they respond to our chemical of interest. Now we hit them with the chemical and see how they respond. But instead of taking static measurements every 15 minutes now, we can measure them with high fidelity because we're just measuring fluorescence, okay? They're labeled now. And so you get now these beautiful curves. These look like simulations, but I kid you not, this is nonlinear response in biology to a new chemical, all right? Full change response, okay? So lastly, we took those chemicals and we actually, we then exposed them to real cabbage plants in a garden. And we collected the runoff from that. And then we actually exposed the runoff to our biosensors. And here's the data from it. You can see that we are actually able to detect with our identified biomarkers using DMD the response to this new chemical. So that is to say, in 3 months' time, we're able to functionalize biosensors using Koopman operator theory. A process that normally took 5 years to do, and a PhD student takes 3 months, all right? And the algorithm, of course, is available online. So should we use Koopman operator theory for biology? Hopefully the answer for you guys is yes. Okay, this is all published in a Nature Comms paper. Dr. Hasnain was the leading author of this. Sorry, it's not Nature, it's just Nature Comms. I was having an interesting conversation with someone about trying to push a little bit further. I think we'll get there, but by the end of this talk. I should note that there is also applications of using this exact library to test 1 minute, right? 1 minute, yes. Okay, so I'm going to skip that then. All right, we can also use it to identify CRISPR targets. I'm just going to skip this theory here. There's some new results here where we've shown that you can do data-driven observable decomposition that allows us to then identify CRISPR targets to modify the growth rates of organisms. So here is from the needle in the haystack, 4,000 genes, 4,000 choose 3 versus 4,000 choose 2. It's a huge number. Pick out the genetic targets that are going to help you increase your growth rate. Every single predicted growth variant matched exactly how it was in real experiment. This is a plot that shows the growth benefit. Everything in this quadrant means it grew faster because of our Koopman-based control. And then everything that's on this side, on this left side here, is something that was predicted to have a growth defect based on Koopman-based control. So, I mean, the chances of that happening just from random chance are like astronomically low. And the fact that it all works on the first try is really quite impressive. We didn't have to iterate on this loop. So I'm just going to finish with this comment, because the next time I see you guys, I hope to present Koopman operators used to study epigenetics, and specifically the fate of cells. So as you guys know, you all have gardens probably, and garden knots in your garden hoses. And the idea here is that DNA is naturally twisting, and DNA twisting actually correlates with cell fate. All right. So we've gotten to the point now where we can actually bind DNA on glass, and we have DNA bound to glass and subject to magnetic force. And when you rotate those beads, you can actually introduce a defined level of a twist. Why that matters is because these twist dynamics actually are quite interesting and happen very quickly. There are spatio-temporal dynamics. Oh, I think I know a method that is really good for spatio-temporal analysis, for extraction of modes, and for figuring out the dynamics, right? So this is work that was done by Kees Dekker's group in the Netherlands. And what we have done at UCSB is create the world's first visualization of this supercoiling process with real-time transcription, meaning with real mRNA expression, polymerase docking. Everywhere you see a green is a polymerase docking. Everywhere you see red, that's DNA that's been stained and labeled. And so you can actually see pictures. If you dissect those pictures, you can actually see the DNA supercoiling knots as large red puncta, and then the green dots are the transcribing polymerases. You can actually track as a function of time. The position of each of these things, the knots and the polymerases. So there's a way to actually finally build physical models that correlate with the expression of gene expression. And why this is important is because we've seen in our in vivo data that when you change the constraints on boundaries, it completely changes the fate of cells. 3 different strains that are genetically identical, but if you change the boundary constraints on their supercoiling, changes the fate of cells. So it's a really important epigenetic phenomenon. So hopefully in the future, I'll present to you Koopman operator methods for learning those characteristics. The conclusions I'm not going to read through because I'm out of time. I just want to leave you with a slide on the challenges. I think there's lots of opportunities here, and I'd be happy to take any questions that you have.